This procedure begins with overexpression of the recombinant C-terminus of the purinergic P2X receptor, or P2X2, engineered as a GST fusion protein. Following bacterial lysis, purified protein is incubated with glutathione cephalose 4B beads, and the concentration is read using the Bradford method. Next, proteins from purified rat brain lysate are incubated with the C-terminus of P2X2 receptors bound to glutathione cephalose beads, and protein complexes bound to the C-terminus of P2X2 receptors are isolated via SDS phage gel electrophoresis and excision and are identified by mass spectrometry.
Hi, I'm Harpreet Singh from the laboratory of Baljit Khakh, Department of Physiology at University of California, Los Angeles.
Hi, I'm Sarah Warburton from the laboratory of Tom Vondriska in the Department of Anesthesiology at the University of California, Los Angeles.
Today we'll show you the procedure to isolate and purify the C-terminus of P2X2 receptors. We use these proteins to identify the interactors from the rat brain, and in our laboratory we use these proteins to study the functional consequences on P2X2 receptors.
So let's get started.
The cytosolic domain of the P2X2 receptor consists of the N and C termini. The C-terminus of the P2X2 receptor will be used as bait for the pull-down assay. Also shown is the sequence of the P2X2 receptor C-terminus used for this interaction study. To express the